Genome projects are projects to determine the entire DNA nucleotide base sequence of a wide range of organisms, including humans, and these have taken place over the last few decades. The idea has been to map the DNA base sequences that make up the genes of the organism and then map these genes on the individual chromosomes of that organism. In this way, a complete map of all the genetic material in an organism, which is called the genome, can be obtained. Sequencing. The human genome consists of over 3 billion base pairs. This would have been impossible without the use of bioinformatics in order to make a list of all the base sequences, and it took 13 years. Bioinformatics is the science of collecting and analysing complex biological data such as genetic codes, and it uses computers to read, store and organise biological data at a much faster rate than previously. It also utilises algorithms, which are mathematical formulae needed to analyse and interpret biological data. DNA sequencing. To determine the base sequence of the DNA, an organism uses the technique of whole genome shotgun sequencing. This involves researchers cutting the DNA into many small, easily sequenced sections and then using a computer algorithm to align overlapping segments to assemble the entire genome. Sequencing methods such as these are continuously updated, which along with the increased automation of the process involved have led to extremely rapid sequencing of whole genomes. So now, instead of 13 years, it may take just a few days. The medical advances that have been made as a result of sequencing the human genome are many. For example, over 1.4 million single nucleotide polymorphisms, or SNPs, SNPs, have been found in the human genome. SNPs are single-base variations in the genome that are associated with disease and other disorders. The proteome. Of greater practical importance to humans is not the genes themselves, but the nature of the proteins these genes code for. These proteins are known as the proteome. A general definition of the proteome is all the proteins produced by the genome. However, as a protein is only produced when the gene is switched on, and the genes are not switched on all the time, a more specific definition is all the proteins produced in a given type of cell or organism at a given time, under specified conditions. These are differences in the ease with which we can determine the genomics and proteomics of simple and complex organisms. Determining the genome and proteome of simpler organisms. The first bacterium to have its genome fully sequenced was Haemophilus influenza in 1995. It contains 1,700 genes comprising 1.8 million bases. The genomes of thousands of prokaryotes and single-celled eukaryotic organisms are currently being sequenced as part of the Human Microbiome Project. It is hoped that the information gained will help cure disease and provide knowledge of genes that can be usefully exploited. For example, ones from organisms that can withstand extreme or toxic environmental conditions and so have potential uses in cleaning up pollutants or in manufacturing biofuels. Determining the proteome of prokaryotic organisms like bacteria is relatively easy because the vast majority of prokaryotes have just one circular piece of DNA that is not associated with histone proteins. There are none of the non-coding portions of DNA which are typically found in eukaryotic cells. Knowledge of the proteome of organisms like bacteria has a number of applications. Of particular interest is the identification of those proteins that act as antigens on the surface of human pathogens. These antigens can be used in vaccines against disease caused by these pathogens. In the case of vaccines, the antigens can be manufactured and then administered to people in appropriate doses. One example is sequencing of the DNA of Plasmodium falciparum, which causes malaria. All 5,300 genes of Plasmodium's 14 chromosomes have been sequenced, giving us an insight into its metabolism and knowledge of the protein it produces. All this will be invaluable in helping us to develop the elusive vaccine against this globally important disease. Determining the genome and proteome of complex organisms. The success in mapping the human genome in 2003 is a testimony to what can be achieved in mapping DNA sequences of complex organisms. There are around 20,000 genes in the human genome, although this number is constantly being revised down as our techniques for identifying genes improves. The problem in complex organisms is translating knowledge of the genome into the proteome. This is because the genome of complex organisms contains many non-coding genes as well as those that have a role in regulating other genes. In humans, it is thought that as few as 1.5 of the genes may code for proteins. There is a human proteome project currently underway to identify all proteins produced by humans